Okay, next we'll be looking at collagen. Right, I'm sure you have heard of collagen before, right? Before the beauty products and also the beauty in the pot, right? With collagen soup and all that. Okay, so collagen is a protein. It's a structural protein, or rather, it plays a structural support function. Okay, it is the main protein found in connective tissues in animals. So it is found in our skin, our bones, different parts of our body. All right, and it is a very important component of something called the extracellular matrix. Right, so your cells are obviously not floating around freely inside your body, right? So they are anchored by the extracellular matrix, so that your cells can be organized into particular shapes. When the cells group together, they can form the tissues of particular shapes, organs, and so on. So collagen is one of the important components of this matrix. The collagen is made up of a repeating units or subunits called tropocollagen, which itself consists of 3 polypeptides twisted together to form a triple helix. So we will look at the structure in a bit more details later. Okay, so let's look at the primary structure first for each polypeptide involved in the formation of collagen. The sequence, right, the amino acid sequence of the collagen polypeptide, right, shows a particular pattern of repeating sequence. There is always a glycine on every third amino acid, followed by 2 amino acids that are commonly proline and hydroxyproline. Okay, so when we look at the makeup, right, the amino acid sequence of collagen, right, we find a lot of glycine because every third amino acid is a glycine. We also find a lot of proline. There are modified amino acids in collagen, right? So these are amino acids that you don't find in the table showing you the 20 amino acids in the notes, right, in figure 1.3. These are the hydroxyproline that is commonly found, and also there are some hydroxylysine as well. Hydroxy meaning OH group lah. So it's like proline and lysine. These are the normal amino acids added with a OH group. So it becomes hydroxyproline and hydroxylysine. Okay. Then we look at the secondary structure. So each of these polypeptide will be coiled into something called a kinked helix, right? So I show you a picture here, kinked because there are all these abrupt bends, right? So if I just bring up my laser pointer, all right. So look at this one. This would be one polypeptide, right? A second one which is in green, and then a yellowish beige one that will be the third one. So looking at each of them, right, it is kind of kinked meaning there are bends, right, in the polypeptide. So it forms this helical structure that is not really smooth, unlike the alpha helix, right? There are all these sharp bends, right, in this helix. So it's called a kinked helix. Right? So it's certainly not an alpha helix. Whatever you have learned in alpha helix doesn't apply to this kinked helix formed by the polypeptide in collagen. Okay, just take note of that. Then we will jump to the quaternary structure because collagen doesn't have a tertiary structure. Right, it is not a globular protein. In fact, it's called a fibrous protein. Fibrous, like fiber, because it is long and strand-like. Right, so it's not a globular shape. There's no extensive folding of the polypeptides into a compact structure. So there's no tertiary structure, right? So from secondary, it jumps to quaternary. So just now we mentioned, right, collagen is made up of these repeating subunits of tropocollagen. Each one is made up of 3 polypeptides. So one tropocollagen itself is already a quaternary structure. So this is not even the collagen yet. It's just one subunit within collagen, right? A tropocollagen already is a quaternary structure with 3 polypeptides forming a triple helix. So collagen is a very strange protein, right? So just in the first SLS lesson, you learned that for quaternary structure, right, the polypeptides associate together by interactions between R groups, right? But for collagen, it is different. Okay, for collagen, the 3 polypeptides in the tropocollagen associate together via only hydrogen bonds, and the hydrogen bonds are formed between peptide bonds, formed between CO and NH of amino acids found on different polypeptides. Okay, so it's very different from a typical quaternary structure. Yeah. Just now we mentioned, right, every third amino acid in each polypeptide is a glycine. So if you remember what glycine is, right, you can look at figure 1.3, look at the R group table to look at glycine. You will find that glycine is the smallest amino acid. Its R group is just a H. Okay, so when the triple helix is formed, right, in the tropocollagen, glycine will be coiled in such a way that it is found in the interior of the triple helix. So because it's the smallest amino acid, its R group is just a H, it allows the 3 polypeptides to be coiled more tightly together. So you have this diagram in your notes, right? So looking at this diagram, you can see, right, when glycine is found in the interior, right, its R group doesn't take up so much space because it's the smallest. So it allows the 3 polypeptides to be closer together. So if it's not glycine, right, if it is another amino acid with a larger R group, then it will take up more space, right? Then the 3 polypeptides will be like pushed further apart and cannot twist so close together. Okay, so that's the reason why glycine, right, is so commonly found in collagen, and it is always found in the interior of the triple helix. Okay, so this diagram you also have in the notes shows you the formation of hydrogen bonds, right? It's between the CO, right, and the NH, mostly lah, at least, right? CO and NH, right? This one shows you water that is like linking in between, but then most of the hydrogen bonds, as you can see here also, right, is between CO and NH, right? So just focus on these ones lah, the normal ones, right? Okay, then move on. Going back to the quaternary structure, right? So we mentioned tropocollagen being a quaternary structure already, but again, tropocollagen is not collagen yet, right? Just one subunit of collagen. So the tropocollagens will come together to form something called the collagen fibril, right? The tropocollagens will form covalent cross linkages with one another, and these covalent cross linkages is formed between the R groups of lysine residues on adjacent tropocollagens. Okay, so again, this is very special. This such cross linkages is not found in other proteins, right? Because the only covalent bond between R group for normal protein is the disulfide bond formed between cysteine, right? But here we have these special covalent cross linkages or covalent bonds formed between lysines via the R groups, right, of adjacent tropocollagens. So when the tropocollagens come together to form the collagen fibril, right, they are arranged in a staggered manner, right? So you can see here, each block is a tropocollagen, right? So staggered so that it prevents the lines of weakness, right, in the collagen fibril. Unlike if the collagen is not staggered, then you will have all these gaps, right, between tropocollagens. Then the collagen fibril structure will be weaker because there are lines of weakness, but not when it is staggered. Okay, so the tropocollagen associate, right, into collagen fibril, and then multiple collagen fibrils will then also associate to form the collagen fibers, and that's where we have collagen. Okay, so Collagen, as you can see, is formed by the aggregation of collagen fibril, which itself is an aggregation of tropocollagen, which itself is an aggregation of 3 polypeptide chains, right? So you have so many polypeptide chains basically bundled together into this very large bundle. It is very strong, very stable. So we call this tensile strength. So the collagen fibers, right, or collagen protein as a whole, will have a very high tensile strength, which makes it ideal to perform the structural support function. Okay, so just a picture to kind of summarize, right, the aggregation, right, of polypeptides to form collagen. Yeah, so 3 polypeptides to form tropocollagen, many tropocollagens to form 1 collagen fibril, and then also multiple collagen fibril to form 1 collagen fiber. Okay, on the left here you have an electron micrograph of collagen fibril, right? So this is 1 fibril. It shows a banded appearance. That is because of the staggered arrangement of the tropocollagen in the collagen fibril, right? Because of the staggering, right, there are some gaps and then there are some overlaps of tropocollagen. When it is seen under the electron microscope, right, it shows a banded appearance. Okay, so next we will look at how the structure of collagen allows it to perform its structural function. Okay, so it is a fibrous protein, not extensively folded. The out groups, if hydrophobic, are not shielded in the interior of the protein, right? It's not globular, right? So there's no interior, right? It's a fibrous protein. Okay, so that makes collagen insoluble in water. So you can imagine, right, it performs structural function, but if it dissolves, then how does it support anything, right? So it cannot dissolve in water, right? So therefore, being fibrous, being not extensively folded, not having a tertiary structure, allows it to be insoluble, therefore perform support function. For the glycine, right, every third amino acid is glycine in the polypeptide. We have mentioned this already. So this allows the triple helix in the tropocollagen to be coiled more tightly. So the tropocollagen itself is more stable, and therefore the final collagen is more stable as well, increasing tensile strength. Each polypeptide forms a kinked helix, is fibrous, right? So it is long and strand-like. So that allows more hydrogen bonds to be formed between the polypeptides in the tropocollagen, because the hydrogen bonds are formed along the length of the polypeptides, right? So when the polypeptides are not, compact, when they are basically still very long, elongated, then there are more hydrogen bonds that can be formed, right? So the tropocollagen is more stable, again, increasing the tensile strength of the collagen. Between tropocollagens, there are covalent cross linkages that allows the formation of the collagen fibril for the tropocollagens to come together, right, to form the fibril. That also, the covalent bonds, being strong bonds, right, also increase the stability of the fibril and therefore the tensile strength of the collagen. The tropocollagens are arranged in a staggered manner, so again that prevents lines of weakness. We have mentioned this already. That increases the stability of the fibril and therefore tensile strength of collagen. And the aggregation of fibrils into collagen fiber increase tensile strength greatly, right? So it's an aggregation of so many polypeptides, right? Such a large bundle, right? So therefore, this is very strong. Okay, so again, collagen is a quaternary structure, a quaternary protein, right? We've made up of so many polypeptides. But the most important level of organization is actually the secondary structure, because the secondary structure makes it fibrous, makes it long and strand-like, right? There's no extensive folding. So that is what allows collagen to be insoluble. That's what allows collagens to be able to twist together to form tropocollagen and then to form your fibril and your fiber. Okay, so the most important level of organization for the function of collagen is the secondary structure.